cannabis plants have been used by humanity since antiquity. Over 10,000 years of usage for medical purposes, for recreation purposes, for creation of fibers for clothes and material. But today the modern utilization is different because we are talking a lot about using it for modern medicine. Part of the magic in this plant is the constituents, chemical constituents. There are more than 600 chemical constituents in cannabis with potential medical activity. And the production of those chemicals in the plant is very much affected by cultivation conditions. Yet we have very little information of what affects the production of this plant. How much water do we need to give the plant? How is the concentration of fertilizers or light intensity or temperature or humidity affect the production of the therapeutic chemicals in the plant? Yes, there is a lot of information if we look at the internet or other places, a lot of information about effects on the plant, but this information is not science-based. And because this plant now is going to be used for agro-high-tech, for the pharmaceutical production, we need to understand on a physiological level, on an agronomical level, how does the conditions of cultivation affect the chemical makeup and to develop optimal cultivation practices. In addition to cultivation condition, a second major effect on the chemical profile of the plant is genetics. There is a huge variability of cultivars and types of cannabis which vary in the constituents which they have. There is a lack of information about which compounds is required to treat different medical conditions. Which cannabinoids or terpenes do we need, for example, to treat Parkinson? Which cannabinoids or terpenes or flavonoids do we need to treat different types of cancers, and so on. And so there's a need, on one hand, to learn the condition of cultivation that will encourage the development of the chemical makeup that will required, and on the other hand, medical studies to find out which requirements are best for treatment of different indications. So the project that we do in the lab is we study the response of the plant to different type of cultivation conditions. We check for agronomical factors, we check for chemical factors and medical related compounds. We therefore grow the plants in a greenhouse. We then extract the material and study chemical profile. For example, this is the result of some fertilization treatment. So we will analyze the accumulation of maybe nitrogen in the plant and potassium in the plant, but also we will study the effect on cannabinoids and terpenoids and so on, with the goal to find cultivation conditions which are best suitable to standardize the profile, to make sure that when we cultivate the plants in January or in February or in the winter or in the summer, we continuously repetitively get the same chemical profile on the plant, and also that we encourage production of chemicals which are helpful medically. And in terms of genetic variability, there is a huge variability in cannabis. Some of it may be reflected on the size of the plant. Some of the plants are more, the growth is more dense. Some of the plants will grow bigger or smaller. We have different shades on the leaf, different shades to the leaf. This is extraction of one type of cannabis which produces more anthocyanin. It's a pigment in the leaf, we have this is an extraction of a different type of cannabis, which is more typical, that has more chlorophyll, which is a green color of the leaf. And those changes, some of them are being reflected also in the inflorescence, in the flowers, which are then being used medically. So we do a combination of working on different genetics together with different cultivation conditions to optimize the quality for general production as well as for specific health indications.